Well, hello everybody. I'm Nick Nicola, the Assistant Director at the Walter and Eliza Hall Institute. I'd like to very warmly welcome you all to the Institute on this special occasion to hear two public lectures in celebration of Professor Don Metcalf's 80th birthday. Don Metcalf has had a very fortunate life in many ways. One of the most fortunate things that happened to him was in 1954, when the then director of the Anti-Cancer Council of Victoria presented him with the Carden Fellowship. Now, Bill Keogh, who was the director, was a very good man and a very smart man, but he was a lousy lawyer. And when the contract came, there was no termination date associated with the fellowship, which Don has held the Anti-Cancer Council to for 54 years now. And in recognition of that generosity, we thought it was only appropriate that we invite the current director of the Cancer Council of Victoria, Professor David Hill, to act as Master of Ceremonies today. Thanks, Dan.
Well, I thank you very much, Nick. I think in fairness we should say from the then Anti-Cancer Council's point of view and the current Cancer Council of Victoria's point of view, we've wanted to hang on to Don Metcalf as well, and it's been a very happy relationship. I'd like to thank the current and future directors of the WEHI, Professor Corey and Professor Hilton, for asking me to MC this wonderful occasion. My organisation, the Cancer Council, and yours have had a long and a very, very happy association, mostly but not exclusively focused on the brilliant career of Professor Donald Metcalf. I first got to know of and then to know Don around 1964, I think, 63 or 64. Both of us were mentored differently, of course, by that unsung intellectual giant, Dr. E.V. Bill Keogh, who Nick has just mentioned. Bill Keogh used to mentor his proteges in his smoke-filled flat in South Yarra, in a street there, I've forgotten the name of it, opposite Melbourne Grammar, where you would call for an hour or so on your way home from work. And I not infrequently used to run into Don, one of us, either coming or going from those sessions. And I certainly know that, and I'm sure this would be the same for Don, they were extremely encouraging and inspiring meetings. I'm sure that Bill Keogh would join us in the immense pleasure that we all share today in celebrating not only Don's 80th birthday recently, but his enormous contribution to medical science, the treatment of disease, most particularly cancer. Following Bill Keogh, each of the subsequent directors has delighted in continuing that support, as Nick mentioned, of the work through the Carden Fellowship. And of course that support has been amply rewarded by the wonderful scientific advances that have flowed out of the laboratory, the Institute. Don has taken the discovery of colony-stimulating factors all the way from the finding and understanding of this phenomenon in nature through to the clinical, life-enhancing, life-saving applications. Every time I check the number of people who've benefited from this discovery of Don and his collaborators, the number has risen substantially, and the last time I checked the estimate was over 6 million people worldwide had benefited from the therapies that have arisen from the work that was done in this institute. Don is recognised internationally as the father of modern haematology. He's taught and nurtured and weaned and launched a diaspora of distinguished medical scientists around the world who now occupy leadership roles, and one of whom you'll be hearing from shortly after we hear from Don. But first we are here to hear from Don himself. He really does need very little further introduction. I think all of us in the room, Don is well known to all of us and you to him. We're dying to hear your presentation, Don, which is entitled The Hunt for Blood Cell Regulators. Thank you.
Thank you. Thank you, Nick. Thank you, David. Can you all hear me? Yes. The title of my talk is The Hunt for Blood Cell Regulators, and it was felt that maybe the audience didn't understand what was meant by them. So I'll explain as I go along. You're sitting in this building now, and the story I'm about to tell you has actually been going on for 40 years. It was completed after a fashion in this building. It was well in progress in our earlier building, now the Ludwig Institute, but in fact it started in my even earlier abode, the Animal House of the Hall Institute. So it's had a long history. Story is about two sorts of white blood cell. This one, which is called a polymorph or a neutrophil, or I'll call it a granulocyte, and this one, which is a monocyte or a macrophage. Those two cells are what protect you against bacterial infections, against fungal infections. Without them, you are in deep trouble. The problem about those cells is that they're very short-lived. Their lifespan is a matter of days. And so, as you sit here listening to me, you are continually producing millions, literally millions, of replacement blood cells. And these are the two that we're going to be talking about today, granulocytes and macrophages. But you do this accurately. The cells are perfectly formed, and you do it without having to think about it. Oh, I think I'll make another red cell. Oh, I think I'll make another neutrophil. So it does pose a question. How does the body figure this out? The location of this, the stem cells that give rise to these 8 families, is all over our body, in our sternum, in our ribs, in our skull, in our backbone, in our hips. How does it all coordinate? Well, I have to tell you, there are 3 sorts of people in the world. 2 of them are in this room. There are those who think that that's a complicated process that has to be regulated quite tightly by regulators that will stimulate the cell production and regulators that will tend to shut it off. But there are people, and they tend to be the same people who believe the earth is flat, who say, no, all you have to do is keep blood-forming cells happy and healthy, and they'll do their own multiplying without any stimulation. There is a third group who mainly immunologists, who have the notion that the body can count, and if the body feels there are not enough lymphocytes, they will make more until the required number is present. And not all that fixed about how this is done, but the notion that the body can count actually, I think, is wrong. But never mind. So I'm clearly in this camp here, and what started all this off in 1954 was the fact that I had a fellowship and I was supposed to be studying the nature of leukemia. And most of the people in the world at that time were convinced that leukemia was caused by a virus. Viruses cause leukemia in chickens, they caused it in mice, they caused it in cattle. Why not in man? So everyone around the world was focusing on the possibility that there were viruses that caused leukemia. And at that stage, I should say, it was not all that clear that leukemia was actually a cancer, but they were interested in viruses. Now, I'd fallen under the sway of a Hungarian cancer research worker, Jacob Firth, who had been working with endocrine organs like the pituitary or the thyroid, and had documented very nicely that if you overstimulate these organs for long enough, then they will progressively hypertrophy, get bigger, and then they will become actually neoplastic, as long as the excess growth factors are present. And then a further change happens and they're autonomous. They're just frank cancers. So I thought, well, maybe leukemia development is fairly similar. The only problem was nobody knew anything about growth factors that controlled blood cells. Now, I tell a lie slightly. 1906, two Frenchmen, Carnot and de Flandre, published a paper describing how the serum of rabbits that were anemic stimulated red cell production in 24 hours. Absolutely impossible. They were lies. But they coined the term hemopoietin for this thing in the serum of these anemic rabbits. Nowadays, it's known as erythropoietin. It's a genuine regulator, well known to cyclists around the world. And it was, in fact, the first notion that blood cells might be regulated by something. But for granulocytes and macrophages, it was open to any sort of postulate. Until 1964, when Ray Bradley, over in the Department of Physiology of the university, and I had been working on the thymus for some years, and we decided to try to grow leukemic cells from mice, from AKR mice that develop a lot of lymphoid leukemia. The cells, we tried to grow them in agar cultures. So it's a culture of nutrients, but it's made solid by mixing agar with it, just like jello. And the leukemic cells wouldn't grow, so Ray put underlayers of cells underneath in an attempt to stimulate the leukemic cells to grow. And one of the populations he put underneath resulted in this stunning appearance. Colonies were growing of all sizes. Each one of these had about 5,000 or 10,000 cells in it. But the colonies were not in the layer of leukemic cells, they were in the feeder layer. They were colonies of bone marrow cells. They are, in fact, colonies of granulocytes and macrophages. So this was an astonishing finding because books had been written up until that date saying you can't grow bone marrow cells in culture. They just won't grow. They were growing. And what made that phenomenon so interesting was the fact that each colony was a clone coming from a single ancestral cell, and they wouldn't grow spontaneously. You had to add things, tissue extracts or lumps of tissue to this culture to make these colonies grow. And we called that something the operational name colony stimulating factor, logical name, CSF. Sorry that it's also cerebrospinal fluid, but never mind. No, we thought maybe this culture system will allow us to assay for this factor that we hoped, like mad, might be the regulator of granulocytes and macrophages. But we were worried because the conventional wisdom said you can't grow normal cells in agar cultures. You can grow cancer cells, but not normal cells. So we thought perhaps what we've done here unwittingly is transform these normal bone marrow cells to cancer cells, to leukemic cells. That was worry number one. Worry number two was maybe there was a virus present in these various extracts we'd been adding, and we were looking at viral transformation. Another worry was that it was just trivial, that it was just a small nutrient molecule that was a bit short in supply in the culture medium, and you supplied it. What we were hoping was that it was in fact a regulator that controlled the growth of these granulocytes and macrophages. So we spent a couple of years trying to substantiate this possibility. We thought if it's a real regulator, you should be able to detect it in the serum, probably in the urine, because by then people knew that erythropoietin was in the urine, should be in tissue extracts, and more important, if you develop an infection where you need more of these cells, the levels of CSF should rise. So at that stage I was in Buffalo, and we did a big survey in New York State of patients and a parallel study here in Melbourne. And it turned out, yes, when patients have infections of a variety of types, the levels of this CSF that we could detect elevated. But the clincher came from going back to mice and not giving mice infections, but just taking the cell wall of the bacterium, known as endotoxin, and injecting that into mice. And as you can see, within 6 hours, there was a 1,000-fold rise in CSF in the circulation. Astonishing response. So it looked to be worthwhile. A case had been made that this could well be a regulator, and let's try and characterize it and purify it. To do that job, I set to work a PhD student, Richard Stanley, the elder brother of Fiona Stanley, and the starting material we chose to use was human urine, which we knew had CSF in it. It was cheap, and we put buckets around the institute, and male and female staff members had to use buckets. As a consequence, year after year, there was an enormous stainless steel tank here in which 50-litre batches of urine were dialysing, which really is better not thought about. Now, what was Richard doing here? This is trying to separate the CSF molecule, let's call it a molecule, from what else was in human urine, using a column that would separate molecules according to size. And as they came out the bottom, they were collected in a fraction collector, and then it was my job to assay the levels of whatever was in those tubes. Now, what was bugging progress continually was the obvious fact that there was very little CSF present in anything. It turned out to be only one molecule in a million in the materials we were using. And the technology for separating and purifying CSF, or a molecule of this sort, just wasn't up to the job. We didn't know that in 1968, but that was so. Okay, we move forward now to 1976, I think. Is Tony here? Was it 76?
Yeah, 1977.
Anyway, Tony was recruited from the Weizmann Institute. You'll notice he's using mouth pipetting, which nowadays would be frowned on. I've never met anyone who actually came to any ill harm by mouth pipetting, but never mind. And Tony tried a variety of separation manoeuvres based on size, on stickiness, on charge of the molecule. And very shortly thereafter, he was joined by a second Queen Elizabeth Fellow, our introducing chairman. Interesting, both products of Sid Leach's Department of Biochemistry in the university, both Queen Elizabeth Fellows, no mean feat. Now, by this time, we decided that human urine was boring as hell. It was only stimulating macrophage colonies. They weren't very nice looking. Richard Stanley had gone to Canada and completed the purification of this molecule, which we call macrophage CSF, MCSF. Richard calls it CSF1 because he said that he discovered it and purified it first. Now, in the meantime, we'd found that a much richer source of much more interesting CSF was mouse lung tissue. If you inject a mouse with endotoxin, you get an enormous rise in CSF in mouse lung. So the idea was inject a mouse, take its lung out 3 hours later, culture it for 48 hours, and then harvest that fluid. And that had a lot of CSF in it. Nice, simple job. Our poor, struggling technicians used to do batches of 800 mice at a time, all intravenously injected, all painfully opened up and producing CSF. By that time also it was pretty apparent that there was more than one CSF in lung-conditioned medium. So we split the empire. Tony proceeded on purifying what we call Gm-Csf because it would stimulate both granulocyte and macrophage colonies. And Nick had the job of purifying what looked to be a lousy little molecule because the colonies were tiny. I thought they were dead for years, but they were granulocytes. So this became known as G-Csf. So the world was split, Gm-Csf, G-Csf, both using the same raw material. Progress wasn't made until the late 70s with the introduction of this sort of equipment, high-performance liquid chromatography. Why was that important? Well, this is a table of the purification of G-Csf that Nick achieved with me. It took us 5 years to do it, but you'll notice here that the enormous leap in purification only occurred when high-performance liquid chromatography was introduced. You'll notice that it was a lot of hard work. 120 batches were made. They weren't all processed by these various methods. 250,000 cultures were done to assay the material. I can say 250,000 mice were also used to generate the material. A hard slog. So by early 1980s, 82, 83, it was possible to draw a diagram like this and say, look, what controls the production of granulocytes and macrophages are 4 glycoproteins, choline-stimulating factors, and it all looked a bit redundant, a bit excessive. There was one that was mainly a stimulus of granulocytes, one that was mainly a stimulus of macrophages, and 2 that could do both jobs. But that's the way the body seemed to be set up. Now, I used to hate giving lectures at this time internationally because it always caused trouble. You would say, this is what we've learnt about the CSFs. They're strange things. They can be humoral, just like insulin is a humoral regulator, or they can be actually displayed on the membrane of cells. So a blood cell has to make contact with a cell displaying CSF. And unlike hormones that just come from one tissue, every tissue in the body and probably every cell type in the body can make one or other CSF if provoked. And that was strange. But the thing that people really objected to was this. It was quite obvious that CSFs weren't simply proliferative stimuli. They kept the cells alive. Now, as soon as you showed that, the flat earth people said, see, we told you. All you have to do is keep the cells alive, they'll divide. And it took a lot of hard biochemical work, mainly done by Chuck Scher's group, to document that the CSFs actually had to trigger and initiate a very special set of biochemical events before the cells would divide. people also objected to the fact that the CSF seemed to make cells mature from an immature cell to the mature cells in the peripheral blood. Nobody much argued with the fact that they could activate the mature cells and make them work harder. Now that caused all of us a problem. How could one molecule do all those different jobs? Because it would require an action in many parts of a cell. Nick, when he'd finished Purifying G-Csf, spent a lot of time cloning receptors for the CSFs. On every cell that responds, it has special molecules that allow that cell to respond to a CSF. So the CSF molecule's here in the little brown spot, and there are 2 receptor chains in principle. This is a 1980 diagram, and we wouldn't trust any particular fact shown on it. But the principles are still okay. The way a CSF can initiate many different responses is because there are different regions in the tail of the receptor sticking inside the cell that initiate different signals. So there's a geographical separation. Now I see Bill McKinstry sitting in the audience, so I'll show one of his group's figures from Cell, a recent figure, showing you what a CSF receptor actually looks like. And it's a horrible looking thing, Bill, I'm sorry to say. There's a CSF molecule here, a CSF molecule there. There are actually 4 receptor chains and then a doubling of that 6. So there are 12 chains in the receptor. Very complicated, and that's not even showing yet what's inside the cell. Okay, let's go back to CSF. We were the only lab in the world that had the 4 purified CSFs. We did a lot of clever work with cell culture and the CSFs, but we were actually very depressed. This is what a CSF molecule actually looks like. It's a long polypeptide chain, folded very precisely in 4 alpha helices, stitched together in 2 exact positions, and 2 active faces on the molecule. Everything must be exactly so, or it won't interact with the receptor. It's too complicated to synthesize. People tried. They got CSF-like molecules, but they had terrible activity. It would have taken us 250 years, our whole team, to get by extraction enough CSF to treat 1 human. We figured that wasn't the way to go. The only alternative was to find the gene that coded for that CSF, put that into bacteria or yeast, and mass produce it in fermentation tanks. Everyone says, yeah, that's straightforward. Early 1980s, very few genes had ever been cloned, and there was not much known about how you got expression of them. So it was a tour de force that Ashley Dunn and his group, by now Tony is the director of the Ludwig Institute, Ashley and Nick Goff masterminded our attempts to clone the gene for Gm-Csf. It was no mean feat. The referees of our grant in Washington said, we calculate, I think there might be 1 CSF gene in your material, but go ahead and try. And in fact, there were 2 halves, and they had to be stitched together by Nick. Meantime, Anne Kelso, in our biological half of the team, had come up with a cell line producing bucket loads of Gm-Csf, and it was much easier later to actually pull out intact genes. So this is 1983-84. Within 3 years, all of the 4 CSFs, mouse and human, had been cloned, and the gene for erythropoietin had also been cloned. This was a golden age of molecular biology. So, where next? Where next was Tony having to go around the world trying to interest biotech companies or pharmaceutical companies in expressing these CSFs and mass producing them. Nobody had the slightest interest in them. And eventually he was able to persuade a company in Geneva, Biogen, to try to express both IL-3, or multi-CSF, and Gm-Csf. And 2 years later, this bottle arrived with bacterially generated mouse Gm-Csf. We are 20 years into this project. We've never had enough material to inject into a single mouse. And here it is. We didn't know what dose to use. We didn't know what route to inject it. We didn't know how often it had to be injected. So we had to spend some of our valuable material figuring that out in tests like this. This is what happens when you inject it intraperitoneally and you measure the levels in the blood. They go up and they go down quite quickly. So how often do you have to inject CSF if you know it has to be constantly present to stimulate the cells? Well, we settled for 3 times a day, which meant that in rotation people had to come in at night time, catch the mice and inject them. And we chose intraperitoneal injections because otherwise it would be too difficult. So we're now up to 1986, and it's possible to draw graphs like this. This is what happens when you inject increasing amounts of G-Csf into mice. According to the dose you inject, you can increase the levels of neutrophils almost without let. In other words, the material works in mice. Now, I thought it would take years and years between successful mouse tests and successful human tests. I don't know what I thought. I thought there'd be a lot of toxicology studies done in monkeys, and I didn't really think it through that human CSF won't work in a monkey, so it's pointless injecting human CSF into a monkey. And in fact, the human trials were started within 2 years of the first mouse trials. And they worked fine. Oops. We are having a little bit of electronic. Doesn't want to show that. 1, 2. She does not want to show it. It's an important slide. There we go. This is one of the trials done in Melbourne Hospital, injecting 1 dose of CSF for a number of days into patients, showing that it, as in mice, works fine and it elevates levels of neutrophils. What was very surprising to find, however, when we were monitoring the patients in our labs here, was that there was an enormous rise in stem cells in the blood of these patients being injected with CSF. So much so that the levels here are about as high as you can get by sucking bone marrow out of bones. So the clinician said, okay, why don't we just give patients injections of CSF, collect their blood, and use that for transplantation? And that's exactly what has happened. So just to steal Glenn Begley's thunder a little, the present-day clinical uses of the CSFs are just that, to release stem cells to the blood for transplantation and to stimulate the formation of mature neutrophils and macrophages. I'll let him take on the story from there. Now, the critics said, okay, you've discovered regulators, but you can't really find them in a normal person around this room, only when they're in trouble. They're emergency molecules. That's not how the body works. That's not how granulocytes and macrophages are regulated. So to prove that that criticism was wrong, Ashley Dunn and Graham Leishke had to build a mouse that had no gene for G-Csf, a knockout mouse. And that mouse only had a fraction of the number of normal neutrophils and a fraction of the number of immature cells in the bone marrow. was quite clear from this experiment that G-Csf is the major regulator of all of us when we are making granulocytes. The mice, not surprisingly, didn't respond very well to infections and died prematurely. We assumed the same thing would happen when Ashley and Ed Stanley. Sorry. Made a knockout mouse of the Gm-Csf gene, but to everyone's surprise, nothing much seemed to happen in terms of blood cell levels or bone marrow levels. However, the mice were not happy cats, and they developed a curious lung disease, which we then found human equivalents of, and again they died prematurely. The lung disease they had is this, where instead of having nice, clean alveoli, it's full of this pink staining rubbish, which is surfactant, which should have been cleared by macrophages in the lung, but in the absence of Gm-Csf, they weren't working properly. So there were two sorts of answer from the knockout mice. Either you were down on cell numbers or you were down on cell function. The knockout mouse for MCSF didn't have to be made at vast expense. It is a natural mutant mouse that has solid bones, because mice make bones continuously and they're normally eaten away by a population of macrophages, osteoclasts. And this mouse has an extra nucleotide in the one of the introns and is not making MCSF. And it has subnormal numbers of macrophages. They don't chew up bone properly, and surprisingly, and it should have been a warning to us, they couldn't get pregnant readily. Now, these are blood regulators, we think. So what then happened was that other groups started to discover other molecules that would also stimulate granulocyte production. And I have to say that the reason why G-Csf works so well in a patient or a mouse is because it interacts with this molecule, stem cell factor, and it's really the combined action of these two that gives the big response. So one of the general conclusions is that there are a lot of regulators, and they seem to be an excess number, but they're not really excess, because the body design calls for interactions between them, which is not yet being made use of in the clinic. Now, all went well until Doug Hilton arrived as a student, and we were all tooled up to purify another hemopoietic growth factor. And we were using an assay that was suppression of a leukemic cell line. So we called this factor leukemia inhibitory factor. And we purified it in jig time. We cloned the gene for it. We held all the patents for it. And then the wheels started to fall off LIF. It turned out that LIF is the molecule that people had been scratching their heads trying to find that kept embryonic stem cells stem cells. It was the molecule that people at Caltech had been scratching their heads about that regulated signaling from autonomic nerves. It was a molecule worked on in Japan causing cachexia. And there were actions on the liver. It turned out to be the regulator of ACTH production in the pituitary. And if you knock the gene out, knock the LIF gene out, the most obvious thing that happened was that you couldn't get pregnant, because there should be a burst of production of LIF just at the site where the blastocyst implants in the uterine wall. Now, what the devil is the body doing using one molecule for all these different functions? It looks like a disaster waiting to happen. And we don't understand this. We don't understand why many of the things that we were happy to call hemopoietic regulators actually have some other actions. unfinished business. Now, what about this hypothesis, just to end up my tale of woe? Was it wrong? When we cloned the genes, we had the ability to do the experiments. And the first one we did was to put a CSF gene into a retrovirus and infect normal bone marrow cells. These cells began to produce massive amounts of CSF, and they showed excess proliferation, but they never became leukemic. So the theory was wrong, or was it wrong? He then did what seemed to be exactly the same experiment. Put a CSF gene in a retrovirus into a cell line that had been developed in Manchester when I was working there. And now it made a lot of CSF and immediately became leukemic. The difference was that this cell has learned how to self-renew itself. Every time it dies, the two daughters are exactly like the parent. So then Suzanne and Anthony Perkins did the Clincher Experiment, which was to use a retrovirus that had two genes in, one CSF gene and a gene that influenced the cell's ability to self-renew. And lo and behold, very quickly these cells transformed to leukemic cells. So the general feeling is that there are two sorts of changes that a normal cell has to go through to become a leukemic cell. One is to slightly or severely impair this normal asymmetry of division so that you've got an excess of parental type cells. And for some reason, to learn how to stimulate your own growth. Sometimes by making a growth factor yourself, sometimes by having a receptor that's active all the time, sometimes by having a signaling molecule in the cell. Why it has to be auto-stimulation, nobody knows. But if you have these two changes, you get leukemia. So the sort of experiments we're doing right now are based on that sort of idea. This is work done by Lorraine Robb and Stefan Glaser and I. And essentially, a retrovirus is used to put this gene, which is a homeobox gene that should control patterning in a young animal, into bone marrow cells. And those cells, when put into an animal, rapidly become leukemic. Very potent leukemia-inducing system. This is a dissection of what's going on. What's going on is that within 24 hours of putting this gene into early granulocytes, they learn to self-generate and become immortalized. But they're not leukemic. You can take those cells years later, put them into mice, they won't get leukemia. But if you come along now and insert a CSF gene, then immediately you've got cells that are autonomous and you've got cells that produce leukemia. So we now are at the stage of working back through a whole series of genes that are known to be involved in human leukemia, seeing which part of the story they are involved with. So what have I said? Regulators exist, flat Earth doesn't hold true, and they do control hemopoiesis. They're weird, they have multi-organ origin, they seem to have multiple functions, and you appear still to have too many of them, but the body knows what it's doing. Clinically, there are at least 3 that are in constant use and are valuable clinical agents, as we'll hear. And yes, regulators are involved in leukemia development. I have been earning my money for the Anti-Cancer Council. Now, who do you acknowledge? Normally, there's a smallish group of names. This is a story that's been going on for 40 years, and some of the key players I've listed over here. But other key players like the laboratory technicians, the animal technicians, even the communication department are key to all of these things. And as the materials went into the clinic, it became very important that the team of doctors and nurses and even the patients were critical to the progress of this story. What are the 2 numbers in the middle? 441 are the number of papers out of this division on CSF over that 40 years. We're productive. 329 are the number of separate scientists or clinicians who appear as authors on those papers. So it's an interesting example of what can happen when you have long-term funding, when you have people able to work as groups and tackle quite complicated projects. Thanks very much.
So I remember the buckets well. We're going on now to hear from Dr. Glenn Begley, who's currently the Vice President and Global Head Hematology Oncology Research at Amgen in Thousand Oaks, California. Glenn has had, of course, an early career at the Hall Institute. He was a PhD student with Don from 1982, did a postdoc at the National Institute of Health in the US, and rejoined the division, eventually becoming a lab head. And then in 2000, in 2002, I think it was, went to join Amgen. Glenn, where are you?
Here.
There. Glenn is going to talk tonight to the topic, which I will get to, the blood cell regulators in the clinic, research discoveries that have rewritten, what is it?
The medical
textbooks.
textbooks.
Good medical textbooks, wasn't on my sheet. Sorry for that fumbling introduction. Thank you very much, Glenn.
Thanks very much, David. It's a genuine pleasure for me to be here. Actually, more than that, it's an honour. And you've got some sense of what it's like to follow Don giving a seminar. So thank you, Don. Actually, for me, it's great to be back home, and I feel very comfortable being in this lecture theatre, having stood here many times and suffered the torture of questions from Don and other colleagues. So hopefully it won't be like that tonight. But thank you for this opportunity. Now, what I do have to do is get the slides up, and we'll see if we can do that. Amazing. Tony, thank you. You trained me well. So what we're talking about this evening is making blood cells, and this is what normal human blood looks like. And here are the red blood cells. Here are the white blood cells that I'll refer to as neutrophils during this talk. That's just one type of white blood cell. They're the cells that Don has been talking about as granulocytes. And there are tiny little cells here that you can make out. They all almost look like dirt on the slide, and they're the platelets. The work that Don has described has played an important role in understanding how cells are regulated generally throughout the body, and has also taught us a lot about how cancer develops. But that's outside the scope of this lecture. So I'll be focusing solely on the blood cells and not on the more general implications of Don's work. So I'm fundamentally a simple fellow, and this is what blood looks like to a simple fellow. There are three types of blood cells: the blood cells that carry oxygen, the red blood cells, the white cells that fight infection, and you can see this white cell holding a dagger ready to kill the invading organisms, and the cells that go around plugging up the holes, the leaks, and without them we would bleed to death. They're the platelets. The focus of this presentation clearly is on the white blood cells, or the neutrophils. And the cells are made in the bone marrow, as you've heard from Don, and the grandparent of them all is the stem cell. Relaxing here, sitting in its rocking chair. These are cells that are seldom called on to do very much. When they're asked to do, they act, but most of the time they spend relaxing, doing very little. There are cells within the bone marrow that we refer to as the immature cells, or the precursor cells, or the progenitor cells. They're the cells that actually grow up and generate all of the blood cells that we recognize. This slide is a much more elegant display of what's really going on, and is very similar to the slide that Don showed you. At the top we have the stem cells, or the immature cells. And at the bottom, we have the cells that we recognize as the mature cells in the bone marrow, many of which then migrate into the blood. The macrophages, the neutrophils or the granulocytes, and so on. As you've heard, there are 4 main regulators that control this whole process, and they're diagrammed here. So interleukin-3 or multi-CSF acts on very many of the cells shown here in the purple box. Gm-Csf is also relatively broadly acting and acts on all of the cells within this, I'm not sure what colour it is, yellow-pink box. Macrophage colony stimulating factor, or CSF1, acts predominantly on this part of the family tree. And G-Csf, the molecule that I'll be talking about primarily, acts relatively specifically or exclusively on this part of the tree. But the gradation in colouring indicates that these molecules do have overlapping function, and none of them are purely selective. So the focus of this presentation is on the clinical activity of the CSFs. And on several of the slides, as you'll see as I go through, you'll see just how beautiful these molecules are. This is actually a crystal structure of G-Csf. So it all began in Melbourne. The first patient treated with G-Csf in the world was here in Melbourne in 1988. And here surrounded by Doctors Morstan, Fox and Metcalf. We've actually come on a long way since that time. It's really important to understand that without white blood cells, we are all at risk of dying. These cells are really important in terms of keeping us alive and preventing overwhelming infection. Recognizing that, some of the earliest studies that were performed were to try and understand how G-Csf might help patients overcome the effects of chemotherapy, because chemotherapy normally reduces the white cell count. This is one of the early studies published in 1990, demonstrating that G-Csf is able to accelerate white cell recovery after chemotherapy for acute myeloid leukemia. And on the horizontal axis here, you can see the number of patients expressed as a percentage. And here, it's an old slide from an old study. You can see patients that were treated with G-Csf and patients that were treated with control. The way to look at this slide is to go across here and say, for 50% of patients, where does this line intersect for those that received G-Csf compared with those that received control? And then to drop down and see, well, for patients that received G-Csf, that was 20 days after they'd had their chemotherapy. And for the patients that were in the control group, that was 27 days after they received their chemotherapy. So that may not sound like a very big difference, but for those doctors that are in the hospital wards looking after these patients, that's a substantial difference. And this is highly statistically significant, shown by the p-value in the bottom right-hand corner, which means that this result is very unlikely to have been generated by chance alone. This graph is presented differently, but really shows the same sort of information. And what we are talking about here is the effect of white cells in preventing infection. So here, G-Csf reduced febrile neutropenia in lung cancer patients. Febrile neutropenia actually encompasses 2 concepts. The febrile part means fever, so that indicates infection, and the neutropenia means a lack of the neutrophils, the white cells, that normally would fight that infection. So we're now looking at events for patients with lung cancer that are associated both with fever and a low white cell count, a situation that places them at enormous risk for hospital admission and potentially death. Now, the way to look at this graph is to look after the first cycle of chemotherapy, where you can see that for the placebo group, the patients who received the control arm, approximately 50% of them had fever and neutropenia. For the patients that received G-Csf, on the other hand, 70% of those patients were fine. So that's a very substantial difference. This slide also makes another point that although it's over 20, almost 15 years old, many physicians still have not realised. And that is that if you look at this, the drop here after the first cycle of chemotherapy is very substantial and is actually much greater than the steps that follow with subsequent cycles of chemotherapy, something that I'll come back to later and still fails to have been adequately recognised by most physicians. There was another effect of G-Csf to recover the white blood cell count, and that's shown here for patients that underwent a bone marrow transplant. And again, this was a study that was performed by the Melbourne group. Here are patients who are receiving a bone marrow transplant, and you can see that their white cell count plummets as they get the very intensive chemotherapy that eradicates all of the cancer cells and their normal cells. Then you can see that the normal white cells begin to recover here, and this is under the action of G-Csf. This recovery is very prompt and protects these patients from the risk of infection and death. Shown in the yellow curve here is a control group of patients. There was another effect of G-Csf that was really predicted and doesn't actually get a lot of airtime, but for the patients for whom this is important, this is really life-saving. So there are a group of patients that have this rare disease, severe congenital neutropenia. And for these patients, G-Csf is life-saving. So here is a study from 1990 showing that administration of G-Csf at 3 different doses is able to cause the neutrophil counts to recover in these patients. G-Csf given for longer periods of time is able to maintain the white blood cell counts in these patients. This has been enormously impactful, and one of the physicians that looks after these patients is quoted here, and you can read, prior to G-Csf, approximately 1/2 of affected children died during the first year of life. A profoundly impactful disease. Mortality continued through the 2nd and 4th years at 6 to 7%. These patients are now alive out into 20 and 30 years of age as a result of having access to G-Csf. There are not very many of them, but the impact for them and their families has been profound. So all of those effects were actually, if you will, expected. We thought that G-Csf would recover neutrophils. We thought that G-Csf would then be effective for patients who were getting chemotherapy, for bone marrow transplants, and even the rare patients that had congenital neutropenia. But there was a significant unexpected effect of G-Csf. And Don's already alluded to this. This was the action of G-Csf to move stem cells from the bone marrow into the blood. And you can see here, prior to G-Csf and 5 days after G-Csf, there was a 200-fold increase, at least, in the circulating levels of stem cells that have migrated from the bone marrow into the blood. And again, this is a study performed by the Melbourne group. The second surprise was that if you took those stem cells that have moved from the bone marrow into the blood and then used them in a transplantation setting, there was profound hastening of recovery of the platelets, those little cells that prevent us all from bleeding to death. This was completely unexpected. And what you can see on this graph is the probability of recovering the platelet count, in this case, to a safe level, compared with patients that didn't have these blood stem cells reinfused. It's really hard to overemphasize the importance of this result. This has revolutionized the world's approach to bone marrow transplantation, so that now truly bone marrow transplantation is a thing of the past, and it's blood stem cell transplantation that is really widely utilized. This was an enormously impactful result, again, from the Melbourne group. To try and demonstrate the difference here, you see if you go across at the 50% mark, you can see that for the patients that received the blood stem cells, they had, after 15 days, recovered their platelet count to safe levels. For the patients in the control group, you would have to extend this line off the page until you'd reach the same time point. So G-Csf does 2 really important things. it increases the number of the white blood cells, and instead of them walking around with a dagger, they now have a sword and a shield. They're much more effective at killing the invading organisms. When you inject G-Csf, another thing happens, and you find that there is an increase in the stem cells in the blood that have migrated from the bone marrow into the blood, that they can be captured and then used for bone marrow transplant, really blood cell transplants. Because of those actions of G-Csf, we, the world, were really able to test for the first time the notion of high-dose chemotherapy. So for many years, people had believed, as an article of faith, that more chemotherapy was better. We know that chemotherapy is able to kill cancer cells. The idea was if you could give more of it, you'd be able to kill more cancer cells and have improvement in patient survival. G-Csf allowed that concept to be tested. It's shown here graphically on the left. So you can imagine a chemotherapy regimen that's given every 3 weeks, compared with that chemotherapy being squashed down to be given every 2 weeks. So this is the idea of dose-dense chemotherapy, much more intensive chemotherapy being given over a shorter period of time with the hope that it might result in improvement in patient disease-free survival and overall survival. And that, in fact, is what happens. So for the results of patients after 4 years with breast cancer, there is an improvement in terms of disease-free survival and an improvement in terms of overall survival. Again, with highly significant p-values, demonstrating again that these results are very unlikely to have occurred by chance alone. And it's not just true for breast cancer, it's also true for other cancers, particularly in this case, aggressive non-Hodgkin's lymphoma. And you can see that the overall survival for patients who are receiving the intensive chemotherapy, CHOP14 it's called, plus G-Csf, versus the patients who get standard chemotherapy, CHOP21, there is a significant improvement in overall survival and in disease-free survival. But don't just take my word for it. This is what some of the leaders in the field say. Increasing the dose density of some chemotherapy regimens increases disease-free and overall survival. Using G-Csf makes it possible to give the full dose of chemotherapy on time and as a result improves patient outcomes. So G-Csf is a fantastic drug. It's made an enormous difference to the lives of millions of patients. But you know what scientists are like. They're never satisfied. They always want more. And so there were a group of scientists that started thinking, how can we further improve on G-Csf? How can we actually improve on hundreds of millions of years of evolution? And the answer to that is this molecule, PEG-G-Csf, pegylated G-Csf. Again, the fundamental idea for this came from the Melbourne group, and it was an observation that G-Csf was normally cleared by the kidney. So when you take the kidneys out of an animal, there is reduced clearance of G-Csf. It hangs around longer. We also know, based on the work of the Melbourne group, that the clearance of G-Csf increases as the white cell count goes up. So the number of white cells go up. On the surface of those white cells are receptors that Don described, and those receptors clear more of the G-Csf. So there are two fundamental mechanisms responsible for G-Csf clearance. So the idea then was, could we do something to prevent the kidney clearance of G-Csf? And the idea was to make the G-Csf molecule bigger so that it couldn't escape through the kidney. And that's shown here on the right. At the top is the globular G-Csf molecule, a very small, compact molecule. But by adding polyethylene glycol, or PEG, to the end of it, you're able to increase the volume of this molecule such that it was no longer able to escape out through the kidney. And there is only one way that this molecule can be cleared, and that's via interacting with the G-Csf receptor on the surface of the neutrophils. And that experiment actually worked. So if you first focus over here on the right, you can see that G-Csf is normally cleared by the kidney. That's proven by taking the kidney out of animals and showing that there is a difference between the clearance, a difference between these two curves. And the difference is due to the clearance via the kidneys. This molecule, the G-Csf that's now got the polyethylene glycol attached to it, shows the same clearance whether or not the kidneys are present. The idea being that the only way this molecule can disappear is via the receptor for G-Csf on the surface of the neutrophils. With this molecule, it was able to take G-Csf to the next level. And this, I think, is truly a remarkable accomplishment. And from my point of view, this is probably the cleverest drug that's ever been developed. It's a drug that's cleared solely by the cells that it generates. So it's got this inbuilt self-regulating mechanism, unlike any other drug that I'm aware of. And it really works. I showed you a slide earlier for the patients that had lung cancer and were receiving chemotherapy. And I showed you that there was a significant difference in terms of the episodes of febrile neutropenia. That same information from the same publication is shown here. And you can see that the patients in the placebo group, 77% of them developed febrile neutropenia, fever with low white cell counts, and therefore at risk of death with overwhelming infection. The patients that received G-Csf, this was decreased in a highly significant fashion. It couldn't be due to chance alone. This is data from another study that really showed the same result. The reason for showing this slide is to make the point that G-Csf is effective in the context of situations where about 60% of patients are at risk from developing febrile neutropenia. So these are pretty intense chemotherapy regimens where 60% of patients are going to see their white cell count plummet and be at risk. The study that was performed with PEG G-Csf, the larger molecule that's now only cleared by the white cell count, the question was posed, will this molecule be so effective that you can even use it in situations where only 17% of patients are at risk from chemotherapy? Not 60%, but 17%. And in this situation, you can see that the pegfilgrastim was profoundly effective, a p-value where the number is very, very small, indicating this is not due to chance alone, that the pegfilgrastim was able to reduce the risk, in this case, of febrile neutropenia, even for chemotherapy regimens that are very mild, so to speak. And it also decreased the rate of febrile neutropenia-related hospitalisation. So G-Csf is very effective. Pegylated G-Csf is even more effective. It's not cleared by the kidney. It's only cleared by the target cell, and it results in a profound reduction in febrile neutropenia, hospitalisation, and intravenous antibiotic use. And it's, fortunately, safe and well tolerated. So what are the challenges that face us in terms of using G-Csf even more effectively? Despite the fact that this molecule's been around for 20 years and many physicians are well used to using it, physicians still don't use it optimally. And I'll just give you one example of that as an indication of where we have to go in terms of helping people understand the power of this molecule. So this is a study examining patient outcomes from over 1,000 US oncology clinics. I don't have data for Australia, but I would posit that it's almost certainly going to be similar in terms of the practice of oncology in Australia. And what we're looking at here are women with curative breast cancer, adjuvant treatment for breast cancer. This is a really important disease to make sure that these women are treated properly. And what you can see is that a proportion of them are getting less than 85% of the dose they need to get to give them greatest chance of curing their breast cancer. 55% of patients, the total number looked at is almost 20,000, 55% of these women with curable breast cancer are getting less than 85% of the prescribed dose. It's worse if you're a little older. You're even more likely that you won't get the dose of chemotherapy that you need. And the reason for this is because patients are not getting their G-Csf with the first cycle of chemotherapy. I showed you a slide earlier where the biggest step down was with the first cycle of chemotherapy. Physicians are used to the idea that toxicity is cumulative and it actually gets worse and worse with each cycle of therapy. And there is some truth in that. But for this effect, it's actually the first cycle that's the worst. And physicians too often withhold G-Csf from the first cycle of chemotherapy and they wait till the second to introduce it. And that's the reason why patients are not getting full dose of chemotherapy. It's not just true for breast cancer, it's also true for lymphoma. So G-Csf has been incredibly effective at improving the outcome for patients in terms of chemotherapy-induced fall in their white cell count and hastening recovery from bone marrow transplantation. But the sort of understanding that Don and his group has given us extends beyond there. So one of the lineages on this slide here are the cells that we call megakaryocytes, and they give rise to the tiny platelets in the circulation that prevent us from bleeding to death. And in 1994, Don wrote this article that heralded the arrival of thrombopoietin, the key regulator that drives the generation of megakaryocytes. Last year, two companies, Amgen and GSK, had a molecule approved that takes advantage of that pioneering work. And this molecule is used to treat another relatively rare disease called chronic immune thrombocytopenic purpura. This is a disease where patients have bleeding into the skin as little hemorrhages or bruises, bleeding into the gums, and in the worst situation, bleeding into the brain. And this white area here is blood in the brain because these patients don't have enough of those tiny platelets to plug up the leaks. When patients receive Nplate or Remiplastim that's stimulating the generation of megakaryocytes, you can see that overall there is a dramatic increase in the number of platelets in their blood compared with the patients who received the placebo or the control. And that's true whether or not they've had their spleen removed. Splenectomy is commonly used to treat these patients. So another example of what understanding blood cell regulation has meant in terms of bringing new therapies forward for patients. There are two other molecules that are on this initial diagram, and Don mentioned them both briefly, Gm-Csf and M-Csf. Gm-Csf already has an established role in terms of regenerating white blood cells, but I think the most exciting potential areas for Gm-Csf are still to be discovered. Gm-Csf therapy is currently being investigated in cancer immunotherapy, trying to stimulate the immune cells to recognize the cancer as foreign. In Crohn's disease, a disease where there is inflammation in the intestine, and in wound healing. There are also a number of groups that are very interested in blocking the effect of Gm-Csf, and that's being explored in a number of different diseases. Similarly, with M-Csf, there is enormous interest in trying to block the effect of M-Csf, hoping that that might have a role to play in treating cancer patients, patients with arthritis, and patients with atherosclerotic heart disease. So the title I used was Rewriting the Medical Textbooks. This probably is the Bible, or one of the Bibles, for people that are in the haematology area. And this is the 2006 edition of Williams Haematology. The authorship list here are some of the key leaders in the haematology world. The first edition of this book was published in 1972, and under the heading of granulopoiesis, it actually mentioned colony-stimulating factor. There was only one, and it devoted an entire page to CSF in 1972. The 7th edition in 2006 actually has an entire section to granulocyte colony stimulating factor, and you can read the list there yourself, and another entire section to granulocyte macrophage colony stimulating factor, and another section for MCSF. So I think this is objective evidence in terms of rewriting the medical textbooks. So I work at a company, and one of my colleagues was joking this afternoon asking if I'd been programmed to give the talk appropriately. One of the things that we do have to do is make sure that the presentation like this is seen by our other colleagues and that no one has any objection to the sorts of information that we're going to present. When I spoke with the head of the Neupogen Neulasta program, Dr. Linda Drayling, this was her unsolicited comment. So the first patient was treated in 1988 at the Royal Melbourne Hospital. 4 years later, approximately 300,000 patients had been treated. 1995, we crossed the 1 million patient mark in terms of number of patients treated. And for the last few years, it's essentially been 1 million new patients a year treated. As of November 2008, almost 9 million patients have been treated with G-Csf. Incredible. 4 decades of research, almost 9 million patients treated, new paradigms for cancer therapy and bone marrow transplantation. Speaking to the importance of this molecule, G-Csf has become the standard of care for cancer patients. It's no longer ethical to run placebo-controlled studies that do not have G-Csf. This has become the standard of care. It's forever changed the practice of medicine and continues to dramatically improve the lives of cancer patients. Don, on behalf of the 9 million, congratulations, thank you, and we salute you.
Thank you very much, Glenn, for that wonderful presentation and for demonstrating how out of date I am on the number of patients, cancer patients treated. It really is very, very difficult to keep up with the impact of this discovery. It's now my pleasure to, there's 3 treats tonight. We've had 2 of them. The third is a briefer presentation from Mr. Drew Berry, who's going to give an overview of the CSF animations. So it's going to, Drew will come to the mic and give us the slide and picture show. Thank you very much, Glenn. No, Drew.
Okay, well, thank you. It's a great honor, my goodness, to be up here speaking on this occasion. I'm been asked to speak briefly, very briefly, about the commemorative booklet and the DVD-ROM in the back that you're all going to take away tonight. I'm sure you've all had a chance to browse it. It's full of essays and a great biography about Don and many thoughts about his life and career and the impact that his work has had. I'm just going to briefly speak about the DVD-ROM in the background. Actually, can we switch over to... Thank you. Yeah, there's a bunch of movies on the DVD-ROM for you to view when you're next in front of your computer. The first one is a 2-hour interview by Dr. Max Blythe, the gentleman with the fine mustache there on the left. He was invited over by the Australian Academy of Science to interview Australia's great scientists. And it really is a great interview. I certainly learned a great deal from that interview. Particularly, what impressed me was it really gives you a sense of the work, the hard work, the focus, the courage, and the dedication that's required to really do something that is truly great, something that is worthwhile, something that will have an impact on society for the long term. And certainly Don's work and his attitude towards what he does has done that. So it's a great interview. The second video is of a project we worked on a little while ago, which is Don's Atlas of Mouse Hematology. And when I told Don, I asked him, would you mind if I put this on the DVD-ROM for this occasion? And Don said, of course not. There are 50 people at WeHi that will do well to see this video. And I was a bit confused by that because there are at least 600 people at WeHi. So anyway, I'm going to play a little excerpt. It's a 20-minute video. It's designed for DVD, so if you do find great use out of it, let me know and I'll send you the DVD version. And I'm just going to play a little brief excerpt to give you a sense of what that video is like. In order to do this project, what we did was we set up a number of cameras, one that is mounted directly on the microscope so you can see what Don is seeing down the microscope. Then we also set up a camera so you could film Don as he was talking, and another camera, which you'll see a screen pop up, which is of a video we set up so he can point to the cells as he's talking. So yeah, this is, let's make sure we got audio is okay. Hopefully it's okay. We'll see.
Here we are, lovely monocyte. You'd kill to have a monocyte like that. What's different? What's different? Nucleus is not round shaped. It's bean shaped or U shaped. Cytoplasm is not so small that you can hardly see it. You can see it around there. It's a bluish color and it's faintly granular, or usually described as being like a piece of ground glass. Now we go to the hard one, bone marrow. This is the one that people really don't like to work with. Now they reach for the fax machine to tell them everything. What's the problem? There are 7 different cell populations in the bone marrow. Fax can't tell you that for a start.
So I think we've captured Don with that video. And finally, there's a few animations on there. They're a mixture. There's some that I did about probably 8 or 10 years ago, which I set out a 6 to 8 month plan to build a number of animations that would explain what Don has discovered. And then about 2 months into this project, and I had a lot more work to go, Don makes another, he got another award, major award, and the 730 Report wanted to interview him, and Catalyst was flying, or was Quantum back then, flying in to interview him, and we had to wrap it up in 2 weeks. So I started off well, and then it got a little bit scrappy towards the end. So Etsuko Uno, who's now working with me, had to go at improving those bits that were scrappy. And so it's a mixture of some old and new work. And she's done a wonderful job. Unfortunately, she couldn't be with us tonight because she's overseas at the moment. Yeah, I'll play and talk my way through CSF. Now, the idea of these animations is that they're supposed to support journalists when they're talking about the science, as well as school teachers when they're presenting to school students. So they're reasonably quick, and that's kind of the idea. All right, so the opening shot here is of the G-Csf molecule sitting in water. It's really a useful animation for titles, that's about it. But this is a white blood cell. This is somewhere in your body. There's a bacterial infection. And this cell is spraying out this G-Csf, and so are the other tissues. That G-Csf then travels to your bone marrow. This is the first of the Etsuko's animations. The red regions are the key areas of bone marrow in your bones. So we'll now focus in on that bone marrow. The G-Csf travels through the blood into the bone marrow and stimulates the progenitor precursor cells to then start dividing and multiplying and then maturing into a white blood cell army. So when we pull back out, we'll see these multiplying cells forming these colonies in the bone marrow. They then mature into white blood cells, and then the white blood cells and also stem cells migrate into the blood. So here are the white blood cells traveling in the blood, following the scent of G-Csf to that bacterial infection, and also stem cells go into the blood, which then can be harvested for transplantation. And here's the white blood cell army arriving, clearing out the bacterial infection. And of one other animation, and then I'm done, and that is of this one, which unfortunately Don showed a slide earlier that's of recent work, and this one's kind of now not so right. But anyway, it was right about 8 years ago. What this is a molecular. This is my first molecular animation I did, of a high-end one, which is this is the molecular view of the surface of a progenitor cell. This blue bean here is a receptor, which is like an antenna that's listening or smelling for the G-Csf, very specific. And we'll go ahead and play. So this is a crazy molecular world. You'll see a yellow molecule flying in. That's the G-Csf. And it docks here on the receptor. And it turns out you need two of these receptors then to get together. And when they do, the signal then goes into the stem cell or into the progenitor cell. And we'll pull out lots of the signal coming in, lots of G-Csf. The cell then starts to divide and then mature into the white blood cells. So this booklet was really a team effort by everyone in communications, particularly Simon and also Brad Allen. So take it away, and when you're ready for some more Don, there you go. Thank you.
Well, thank you very much, Drew, and thank you on behalf of everyone for this lovely gift to take home tonight. That's going to be a real treat. Obviously, communication of concepts to the broader community is tremendously important in this work and all our work, and it's an absolutely outstanding aspect of the Hall Institute that you have such a highly developed capacity for communicating this information. And it's work that's really, of course, good for the Institute in my opinion, but also very good for the whole medical scientific community, that it's available to spread the word and help interpret the importance of the work to the broader community. So and my final job is to thank all our three presenters for an absolute treat tonight. They've been wonderful presentations, entertaining, informative, inspiring, and just wonderful all around. So perhaps you'd all join me in a final round of applause. Now, those of you who need to leave now should go to level 1 to escape from the building. And again, thank you very much to the director of the Institute, to all of the staff of the Institute for turning on this wonderful event, and to those of you who've travelled from great distances to be here, particularly our second speaker. Thank you.